So we are living in a tremendously exciting era for cancer treatment and the options that are available for patients. And despite a great deal of therapeutic advances over the past several years, even decade, the reality is that there's also a great deal that is still left for us in the treatment arena. In particular, we think that recalcitrant cancers are representing a very high percentage of patients that are still suffering from the highly aggressive, highly deadly form of many cancers. And so when you look at the survival of patients, many patients, unfortunately, less than half of them achieve a 5-year survival. So with that in mind, cell therapy is actually opening the door to a number of opportunities to actually bend that curve of survival. And many of us are very familiar with recent advances in the realm of T-cell therapy. Now, what we've learned, unfortunately, is that there are limitations, but at the same time, there are opportunities with the current approaches based on T-cell therapy. And so these approaches can be really broken down into maybe 3 categories. Of course, there's an opportunity to improve the efficacy of existing products. The other is there's an opportunity to decrease the toxicities associated with these products. And thirdly is how do we get the products faster and much cheaper to patients? Efficacy, toxicity, manufacturability are the 3 pillars that NK cells attack and are transforming. Cytovia is on the cutting edge of addressing each of these pillars, and we'll be happy to tell you more about each one of those and how we'll be able to improve the efficacy, reduce the toxicities, and make these products more readily accessible and at a lower cost than what's currently available through other avenues. There are currently 3 opportunities to improve cell-based therapies products. One of these opportunities is on the manufacturing front, and this is where iPSC-derived NK cells can really transform the current landscape in terms of manufacturability, both as far as ease of manufacturing, supply chain, and also cost of goods. So again, manufacturability is a huge opportunity for iPSC-derived NK products. And in particular, when we think about that, we think about the ease of manufacturing, the speed at which you can manufacture the product, as well as the cost of goods of iPSC-derived NK cells. So that's on one front. The other one is on the toxicity, and let's roll that into also the efficacy. So the overall therapeutic index of iPSC-derived NK cells. Because of the power of iPSC and the ability to genetically modify iPSC-derived NK cells, one is then able to wire in or take out different proteins that are actually responsible for either the toxicity or the efficacy of a particular product. So you're really in an era of personalized medicine, tailoring the product to the specific tumor type. This is quite unique, and this is where the power of iPSC-derived NK cell really surpasses anything that's out there.
NK cell therapies are a largely untapped area. We know that many cancer patients do not have functional immune cells, and this is where NK cell therapies and other cell therapies can have the greatest potential. As opposed to T cell therapies, NK cells have the potential for minimal to no off-target effects with great potential for efficacy. What's really exciting to me about the technology that Cytovia is building is the opportunity not only to deliver NK cells, but to direct them specifically to the tumor through engineering these cells to be both activated against tumor-specific antigens and to have persistence. There are 2 very important decisions that Cytovia took. Number 1, to focus on iPSCs as the source of NK cells for our products. And number 2, to bring not only NK cell therapies into the clinic, but also to bring NK cell biologics or NK engagers, which together with cell therapies have the potential to boost the immune response and provide long-lasting anti-tumor immunity. iPSCs have the potential to become an off-the-shelf NK cell product that can be manufactured on a large scale with the potential for a safe and effective product that could be administered to many different cancer patients on an outpatient basis. The pipeline of products in Cytovia's portfolio is broad, with potential to benefit across multiple indications with patients who suffer from both liquid or solid tumors. Our board of directors and scientific advisory board is made up of scientific, clinical, and drug development experts in immunology, oncology, and with a high level of expertise in NK cells and in gene editing. The recent addition of Michael Caligiuri to our SAB speaks to the quality of our science and our pipeline. Dr. Caligiuri has made many important discoveries in the field of innate immunity. For example, he and his group at City of Hope recently discovered that PD-L1 expression on NK cells in patients with AML was a key determinant as to whether a patient with AML responds to chemotherapy. These studies have enormous implications into how we engineer NK cells as therapeutics. And of course, this key finding also opens up avenues for combining engineered NK cell therapies with checkpoint inhibitors.